conference with an 8-minute warning, and usually I practice the talk at least one time. I was only testing my talk to make sure the slides worked, and then it changed to you're going to present in a few minutes instead. So hopefully this won't be too bad, and I won't ramble too much. Fortunately, my 20-minute talk will probably fit in the 1-hour slot that Jose had, so it's okay. So I'm going to tell you how we're looking at complex assemblies, which I admit is sort of a generic title, and everyone here could say, yes, we're all looking at complex assemblies. A few minutes ago it said probing the dynamics of large and complex assemblies, and I realized I really can't say large in this group because everyone else is talking about things that are like microns. So really I should just say probing the dynamics of assemblies is probably the more appropriate title for this audience. So I'll tell you one example of how we're using simulations to look at molecular level processes and understand how various components work together in them. So for those of you who know me, most of my independent career has been focused on the ribosome over here on the left, where we've been working with structure-based models or SMOG models to try to look at various conformational rearrangements. I'm not going to tell you anything about that today, other than to say that the work that we did on the ribosome sort of inspired what we did with the spike protein, which is on the right here. So of course everybody's familiar with the SARS-CoV-2 virus, and you've probably seen pictures of the spike protein all over the place through the years. But just to give you a little orientation of what you're looking at here, it's a slightly different picture than you may have seen. This is a structural model based on a cryo-EM reconstruction, where you have this transmembrane region on the virus. You have this S2 domain, and then you have a host membrane. And what this protein has to do is it has to reach out and grab the host, and then pull the viral membrane and the host membrane together to allow for membrane fusion. And by letting these two membranes fuse, the virus can then infect the cell, and that's why we're very interested in it, because this is the essential step that a virus must execute is infection. So what we'd like to do is we'd like to understand how it undergoes this massive conformational rearrangement and see if we can get any insights into that process. So sort of the spirit of what we do, we all have the same challenge in this community, that we have large, slow processes, and a lot of us are physicists as well, so I don't really need to explain the way a physicist thinks, but I'll still take a second. So some people, when they say physics-based approaches, what they mean is we put in every possible detail that we can, and we inspire, it's based on quantum calculations or some sort of renormalization or something, and that's fine. That's one physical perspective. The other approach is what we all see in our undergraduate classes and in grad school, where we talk about the spherical cow, where we say, let's take the simplest description we can of a system, see how far we can take it, see where it breaks, and then add some additional details. And it's really great that I don't need to explain what a spherical cow is in this audience. When I'm in a biology department, I have to go through the whole description of the spherical cow because I realized everyone looked at me like I was a little bit crazy, and I even had a citation in a paper where I cited this paper. And my experimental collaborator said, "Is this the correct citation? Like this must be the wrong thing. Why is there a cow reference on a spike paper?" And I had to explain to them what it meant. They were sort of cautiously okay with it. And we even had a paper where we put spherical cow in the title. This pointer doesn't work that well. We actually put spherical cow in the title, which I was very proud of. And the author here, Fred, actually was the first author on that paper. We got away with it because it's an educational journal, but nonetheless, it's in a title of a respectable paper. So that's the strategy that we would like to use to look at the system. So here is, again, the spike protein. Again, this is the S1 domain, so you usually see this full complex. And what people are often looking at is they're trying to figure out ways of teaching your immune system. Wow, the pointer does not work at all, does it? Oh, whoops, I see. Oh, my finger was in the way. Okay. This would have been worked out during practice. So what you normally see in literature in the field is people trying to figure out how to teach your immune system to target this S1 subunit. So we're going to look at the steps after this S1 subunit dissociates and other regions are exposed and see if we can learn something about how it works, and maybe that will give us new strategies for targeting this virus. I won't tell you anything about effort 2, but if you're interested, we also have strategies. We're working with experimental groups to try to guide new vaccine development, develop new vaccines as well, but that can be discussed later. So an overview of what this protein does. It can be a little disorienting as I will go through this. Hopefully by the end of the talk you'll have some sense of what I'm trying to show you. I'll say up front that the student who did all the work, Esteban Dodera-Roas, he took about 2 weeks explaining this to me every day, and then I would say it the wrong way. It's like, no, that's not what's going on, because it's just such a weird looking rearrangement. And you'll see that in a second here. So there's that S2 subunit. Here's your S1 subunit. And these are a bunch of glycans because this is a glycoprotein. There are also glycans on this subunit, but we don't show them. So the virus has to approach the host. And when it does, what you've often heard about is the receptor binding domains binding the ACE2 receptor. You probably recognize those terms even if you don't work on the system. So it recognizes the host, and then it figures out how to shed this S1 subunit. So it leaves this S2 subunit exposed, which is actually a trimer as well. And at that point, these cyan regions, these fusion peptides, need to reach out, grab the host membrane, search for it, and then retract them back to the viral membrane and bring those two together. So if you look at these two structures, you might have a little bit of difficulty mapping what is actually rearranging here. So we'll look at that in a little more detail. But that's the overall process of what is happening. So why do we use simulations? Well, the experiments only give us the endpoints, the pre-fusion state with the S1 bound and the post-fusion state with S2 gone and the membranes are already fused. That's all we know are the endpoints, nothing about the intermediates, or at least when we started, that was all we knew. This is about a 20 nanometer rearrangement, maybe 30 nanometers. This is a very large extension of the protein. And what we're looking for are any key elements that may impede this motion or may limit this motion, so potentially we can target those regions. And the idea is that the regions that are exposed during the transition are regions that can't be protected by the virus using glycans, which is very typical that viruses will put more glycans on the surface to protect the protein from being recognized. If you have regions that are buried initially and buried at the end, but exposed in the middle, you can't put glycans in the middle of a protein. You can only put them on the edges of the protein. So if we can find some of these transiently exposed regions, maybe we can suggest new targets for how to recognize this protein. So what is the idea behind our models? A lot of people here actually use these models, so I'm not giving this for you, I'm giving this for everybody else, so you can take a break while I explain. So the idea with these structure-based models, we're using all atom representation, so every non-hydrogen atom is explicitly represented in the model. And what we do is we have a known structure and we simply define that as the lowest energy configuration. And there are many reasons you could justify this representation. Like the works of Peter and Jose, the principle of minimal frustration. If you're looking at protein folding, that would be one justification for this type of limited description where you say the native interactions are dominant and you look at folding. That's fine. We're not exactly looking at folding here, so that argument isn't the same argument that we would want to use. And we use these models for the ribosome as well, and we definitely can't use a protein folding argument to justify using the same model for a ribosome undergoing elongation, right? So you have to ask, what are you going to get with this description? So schematically, what we do is we define the post-fusion conformation as stable, so every interaction formed in the post-fusion state is explicitly stabilizing. We have every heavy atom, so atoms can't pass through each other, the sterics is properly represented. And then by construction, if you were just thinking of this pre-fusion configuration, it will be higher in potential energy because we've defined this as the minimum. So when you start your simulation, we would start it over here in this pre-fusion state, and we use traditional molecular dynamics simulations, Langevin dynamics, and the system has to find its way to the endpoint. Now, if you look at this, just downhill energy landscape, you could say this is going to be very boring. You're telling it where to go, it's just going to fall into a hole, so there's nothing to learn. And that's certainly a possibility. If you apply the model to something that's not suitable, you may just find that it just falls into the hole that you built. But if we're looking at more complex systems like this virus or other machines, other things can happen. So for example, you could have this configurational entropy contribution that will be determined by the flexibility of the molecules as well as the excluded volume of the molecules. How many configurations are accessible as the system is progressing from one point to the other. So that will be dictated by the structure and flexibility. So as long as we've encoded flexibility reasonably well and the structure is properly encoded, which is we're sure about that, then there will be this entropic or configuration entropy contribution that can just come out of the model. What can also happen is you're talking about many things that are very irregularly shaped. These are not simple, spheres, even though I'm saying it's a spherical cow. These are, quasi-spherical systems of many spheres that don't fit together all the time. So it's not really a sphere as a system. Instead, all of these pieces may not fit together just right. So even if you know where you're going, things can always get in the way. So if something gets in the way and you have the excluded volume properly represented, it can pause and get stuck, and you could have energetic penalties where a system has to deform to some extent to allow the conformational motion to proceed. So you put these together where the potential energy is fully downhill, and you can get some free energy profile. This is all just a cartoon, but you can get a free energy profile with multiple transition states, and you could start to characterize what the nature of these transitions is. This would be an entropically induced barrier because of these nonlinear change in the entropy, and this could be a sterically induced barrier as an example. So, of course, we could do this and get nothing, but we got something, so I will tell you more and not tell you this is a bad way of going about it. We actually find that configuration entropy and steric effects can be very pronounced in a lot of systems. So let's get back to our system. So here's the pre-fusion conformation, and there's the post-fusion conformation. So where are all the elements going? Well, you have this yellow region that's going from the middle, and it comes down to the bottom. You have this cyan region, which comes from sort of the side in the middle, goes up to the top. You have this dark blue region here that goes to over there. You have the red from over here that goes there, and dark blue that comes over here. Okay, everybody understands how this works, right? This is why Esteban had to explain it to me over and over and over, because he didn't have a nice figure to show me. Even with the figure, I think this is completely impossible to understand what's happening. So the way I describe this is there's these toys that I don't know if they exist anymore that kids have, where it's like a plastic cylinder-like thing with liquid in it, and the cylinder goes inside of itself. When you hold the outside, it folds over in itself and falls out of your hand, and you pick it up and you keep grabbing it, it just keeps folding, the outside goes in. That's sort of what it looks like. Everything on the outside goes sort of towards the middle. Everything in the middle goes around to the outside. So it sort of flips inside out during this process. And that's the best way I can think about how to qualitatively describe what this motion, this rearrangement is. So we set up our model where we define this post-fusion configuration as the lowest potential energy minimum conformation. And then this is an actual time trace of the root mean squared deviation as a function of simulated time. Now the simulation is a simulation with all heavy atoms explicitly represented. We're just showing the RMSD trace here. So when you start, you have an RMSD of about 60 angstroms. So on average, every atom is 60 angstroms from the final position, which is a rather large rearrangement. And as I said, the 0 is where our potential energy minimum is. And what happens is very quickly, you have this drop, and then you sort of get stuck. And you get stuck for a very long time, or long depending on the time scales that you're used to calling long, but long relative to other states. So you get paused right in the middle here, and then eventually it sort of figures out how to go the rest of the way. So this would be one of these signatures I'm telling you about, where this would be either due to entropic factors or steric factors that are impeding this motion. So right away, this very simple energetic model, you'd say, gives you a very strong signature that we would like to understand. So what does this overall motion look like? So here is sort of this intermediate that we are seeing, where the red domain, this head domain, which was right there. There's this red domain. So we're going from this conformation to the next one I'll show you. There's that red domain here, retracts towards the viral membrane, and at the same time, this elongated helix forms, and there are those fusion peptides sort of reaching out, trying to extend to the host. And what happens is we see that it sort of sticks here for quite a while. So the way we described our trajectories, at least at this point, was we said, how far is this head domain from the viral membrane? And we called that Z head. And then we said, how far has that head moved relative to the transmembrane region? So laterally, how far? And we call that R head. And so here is a probability distribution. It's not a free energy, but we can at least detect signatures of these intermediates still. And this probability distribution is calculated over 1,000 trajectories. So we did 1,000 independent runs and then construct this distribution from that. So it isn't just 1 or 2 runs. These are really reproducible events. So you start right here in your pre-state. And you come here, you get stuck in what we call this cage state, which is basically something like that. It sits here for quite a while, and then eventually it escapes over to the post conformation, which in our case it looks more like this. It doesn't look exactly like post because at that point the membranes are close enough that we actually don't need to continue looking at the rearrangement. We just say at this point the work is done, so we're not interested anymore. And this is, again, something for the biologists that don't know how to look at probability distributions. You start at one point, you get stuck, and then you go over. Okay, so it's probably not necessary for this audience, but I probably would have changed that tonight had I, known I was had a little more time to prepare. So I'm not trying to talk down to you. Okay, so we have this overall rearrangement, and then there was a lot of discussion at the time. This is back in 2020. There was a lot of discussion about glycans, these glycoproteins, and using glycans as a glycan shield to try to evade the immune response. So when we look at it, we see all of these glycans are all over the place. Our model is a sterics-based model, and this is a lot of volume occupied by these glycans. So we said, well, maybe they're having some influence in this process. Can we see if we can learn something about it? So what we find is we have this caged state, which I won't go into the details of how we quantify the time. Just basically, we have a way of assigning the time that it's stuck in this caged state, our caged ensemble. We have our 1,000 events, and we can say how long does each event take to complete, or how long does it get stuck in this state. And this is when you have those runs I was showing you, this is the full system being included. We then took our model and just stripped all of the glycans off. So the model is energetically exactly the same except for the glycans. And our glycan representation are just steric glycans anyway. There are no stabilizing interactions with the glycans. So the only perturbation is changing the excluded volume. And what you get is it basically just never gets stuck. It blasts right through that intermediate and continues on to complete the transition. Okay, so for a physical chemist perspective, or a physicist might say, okay, that's really sort of cool, pieces work together, that's interesting. And then the biologist will say, we don't care because it has nothing to do with biology. So then we had to say, well, is it at all interesting biologically, or is it just interesting from a modeling perspective? So what we did is we said, well, we're not actually explicitly putting in a host membrane. We're just considering where the host would be. So in our runs, we can say, if the fusion peptides extend far enough, they in principle could bind a host. So we can set a distance and say if it crosses this boundary, and then the fusion peptides reach far enough, this could be a productive event that would lead to fusion. So we go through all of our runs, and this D fusion peptide is just the position of the fusion peptides, and there are 3 of them. And we ask, do they cross the host distance? And we can then change this host distance and ask, what is the probability of successfully capturing the host, or at least extending to where the host would be, based on all of these different distances. So when we have the glycans, what we have is at 22 nanometers, it's 100% of the time, every single run extends far enough to reach the host. As the distance gets larger, that probability goes down. But in terms of the glycans, we can then do the same thing for the glycans, and what we find is that there's a lower probability that fusion peptides can reach all the way to the host distance if the glycans are absent. So the idea is that the glycans, in addition to shielding, can introduce this pause, and by holding the head in this position, since it is sort of there's a line here, holding the head oriented in this way naturally points the fusion peptides towards the host for an extended period of time, which could increase the possibility of infection. So there was this mechanistic or biological role of the glycans in addition to the immune response role that they have. At least that's what we put out there. And this movie doesn't work. This is what I was going to fix tonight. So it's a nice movie, but there's a better movie at the end, so it's no big loss. So then I'll go forward, and this is 2021, and then comes around to 2023, and Jose and I are talking, because we did all this together, and he said, oh, let's do some more spike stuff. And I was like, okay, yeah, that'd be fun. I'm a little busy, but we can get back to that. Let's do something. Then about a month later, we get an email from an experimental group at Yale that I actually had never met or didn't even know who they were. I almost ignored the email because it looked a little bit like a junk email. And I was like, okay, I'll read it at least. And I read it, and they say, oh, we have these cryo-ET images, and we found your paper, and it makes a whole lot of sense what you're saying. I think you're actually explaining what we're seeing in our experiments. So what they do is they have this virus-like particles that they decorate with the ACE2 receptor, and they decorate with the spike protein, and then these are the two membranes in this pseudovirus complex, and they can capture it at various stages where the membranes are getting closer and closer. And you can see right here, there's one of the little spike ACE2 complexes, or maybe that's just spike actually at that point. Over here, you would have the spike with ACE2 interacting as well. So they took those images and they made some reconstructions, but if you look at these, they say, oh, here's the spike and here's the viral membrane, but they don't see anything in the middle, so they weren't so sure if they were interpreting things correctly, and that's where they went and looked at our paper and said, oh, this actually does seem to look a lot like what you were saying, and now you may be more disoriented because everything is upside down. I just realized, so they look at the system inverted from the way we looked at the system. So my apologies, I did not give a segue that everything now is flipped 180 degrees. Apologies. So I'm sure you're completely disoriented anyway, so it probably isn't that much of a loss. But they couldn't make any sense of their data, and then they took our movies and images and they put them upside down. And they said, oh wow, it sort of looks like what we're seeing experimentally. And so they reached out to us and we went back to our trajectories. We didn't do additional simulations, and we just said, based on these positions of the membranes and the angles that would form with the spike and the membrane, do we see conformations that are similar to this in our trajectories or not? And what ends up happening is that we actually see it in like 90% of our trajectories. Almost every trajectory will see at least one of these states get populated. Many of the trajectories will see conformations that agree with all three. There were actually a couple other similar reconstructions that they had that agrees with them as well. So more or less the overall sequence that they were seeing overlapped very well with what we had already published. And there's just an illustration of a couple of snapshots from the simulation along with a 3D reconstruction that they had. And there's this higher tilt where the viral membrane is approaching the host, and this is also where the viral membrane is even closer to the host. So when I said they were looking at upside down, we were really looking at upside down because the virus does get pulled to the host doesn't get pulled to the virus. So I'll give them that. Ours were upside down. But we published first so we can get away with it. So of course we were very excited about this because that's like a dream come true, right? You do something that you have no experimental collaborators, you have some wild ideas, you put it out there, and then 2 years later an experimental group comes and says, yep, we completely agree with what you said. Like that's unheard of. For all the people in the audience are probably like, yeah, like I hope that happens someday in my career. Coming back to Esteban, he did not appreciate how significant it was that would happen. So he's like, oh, I'm really busy. I'm like, what is more important than doing this? This never happens. You get to work. And so he refocused his attention and realized this was actually very important, that this will probably never happen in his career again. Well, he's really good. Maybe it will, but it's very unlikely. To the extent that this agreed, I say it's unlikely. So now we have all these trajectories. We can start to say for these different ensembles that they detect experimentally, we can sort of put them in a time order, saying when do we see them in our trajectories, which ones are more long-lived, and so on. So provided sort of a time interpretation to their experiments as well. Then we took it one step further because they had looked at antibodies that had been found in human samples. And there were these two that they were looking at called CV325 and CC25106 that bind the spike. And when we looked at the epitopes for those antibodies, they are both basically occluded initially. And then this is just our original simulation. It becomes highly exposed. The CV3 antibody stays exposed for a much longer time than the CC25106 antibody. So they're color-coded in case you're having trouble following those the excessive list of letters and numbers I'm rattling off. So the main point is the purple epitope remains exposed for a very long time, and experimentally this antibody was more effective at neutralizing the virus as well. So there seemed to be some reason to think that this exposure that we're seeing in our simulations could be interpreted in terms of the efficacy of a potential antibody binding to its epitope on the system. So then to sort of look at how this antibody may be impeding the motion, we then actually finally did a new simulation. So we weren't going to get away without doing any new simulations. So we put in a model of the antibody where the antibody simply binds its epitope. We then subject the system to a pulling force which represented it being tethered to the host membrane and the viral membrane. So it has to exert force to pull those together, so we just applied a force to mimic that process. And the distance here is just basically how close are those two membranes. So in black, you have about 20 trajectories here without the antibody, or the Fab in this case. All of them get to the endpoint, and fusion should be accessible. You put in this antibody, just binding this small, like 8 residue region, and you don't see it happen at all, at least under certain ranges of the force. Of course, if the force is too high, nothing ever completes, and if the force is too low, it can still find its way to the endpoint. But in that intermediate range of about 50 piconewtons, it would just basically pause and halt just due to binding of this small region. So that seemed to be it's all consistent with what they're seeing experimentally, and it seemed to give a nice mechanistic explanation for how this antibody may work. So Peter's in the audience. I usually have to quote Peter, but he's here. He always tells you, don't draw any conclusions from movies. And I always remember that, so I tell people that if you're patient and you walk through all the data and arguments, I'll then show you a movie at the end as a reward, because none of the conclusions are from the movie. This is just, sort of a pretty way of looking at it. So there's our viral membrane at the bottom. Here's our spike protein, and this is the process you usually see, which is that receptor binding domain binding to the ACE2 receptor, and then we will show a reconstruction from the same study that they had. So here's this recognition complex. After that, the S1 subunit needs to shed along with the ACE2. They need to get out of the way, exposing this S1 subunit so it can then reach out, and here's an actual simulation where those fusion peptides are at the tails here, and they're just sort of searching for this host that is sitting extended out over here. This refolding process was actually just a cartoon, so that's why it doesn't agree with the previous slides, but the point is it extends far enough to bind to the host, and at that point it could pull the virus back towards the host. And it will pause at certain points, and it will show you the reconstructions from the experimental studies as well to illustrate the overall process. Not all of the intermediates, just a couple. So there's one of the reconstructions, and that's just a pause in this movie. And then it will continue. That was the mid-tilt intermediate, and then it will be more highly tilted. And there you see again, it sort of sits right nicely in that density, except for these disordered regions, which you wouldn't expect any image of anyway. And at the end, they insisted that we hide the virus at that point because we have no actual fusion in the simulations, and they didn't want to make it look like we were claiming it. So, but that is the post configuration. So then we'll show how this Fab could work as well, or at least how it works in our models. So we have this conserved binding site. This is conserved across the variants, across many variants of concern. So this could be provide broad protection against this virus. So there's the antibody bound, and it basically just prevents this region from back zippering or refolding to form this six helix bundle. And by preventing that final process, it just can't utilize that energy to pull the membranes together. That's the idea. So it will just sort of sit here for a while. So that's the idea that's out there, that this highly conserved epitope may be targeted and provide broad protection against many of the variants of concern through a mechanism of this fashion. Probably isn't exactly molecularly perfect, but the overall picture we think is pretty good. So all of our tools you've seen can be applied to lots of different systems. We've even done some test systems where we did a 4 million atom chain of DNA and just put like 1,000 histones in there just to see if the software would work. It works. I'm not doing any studies with that, but if you want to, go ahead. You can do things like hundreds of ribosomes in a simulation. It runs just fine. You can, like the HIV capsid, you can put ions in as well. And you can just sort of create all sorts of different types of models. So if you want to use our software, talk to me. It's all freely available, but it's been really thoroughly tested on many complex systems, so we encourage people to play with them. With that, I'll thank everyone that I've collaborated with. I have all the wrong names on this last slide because I had not actually looked at this slide yet. So I didn't say anything about the ribosome. Esteban did all of the work on the spike protein. Everything was in collaboration with Jose and the Motz lab over at Yale. Of course, thank the NSF for funding CTPP for 22 years and funding me independently as well. So none of this would have been possible without NSF support. And the AMD HPC fund has been really great as well. They've been giving us hardware and cycles over the last few years, which has really enabled all of this work. So with that, I think I'm sort of on time, so not bad for a practice run. So thank you.
We have plenty of time for questions.
Hi, Paul. Very cool. I have two questions. I remember when they were designing this pre-fusion, making that stabilize, they had some prolines or something like that.
They had some what?
They had some prolines, or they had to mutate certain ones to stabilize the pre-fusion state. Is that in your model, or is this the wild type?
This is the wild type.
The wild type,
Yeah.
okay. And did I miss, there were usually you need trimers, right?
Yes, these are trimers.
So this we had the
Right.
trimer.
The S1, S2 complex is a set of trimers, then it's cleaved, and then the S1
You
trimers
simulated
dissociate,
with all of
and
that.
then we leave the S2 trimer for our simulations. Yes.
Okay.
Thank you. Great talk. Paul, how do you add the glycosylations on the protein that you showed?
How do we add them in our model?
Yes.
In our case, we just, we list what all the atoms are, where all the bonds are, and we tell it, make it not attractive, just give it excluded volume. So it's basically a file that we just define the rules for constructing any element that we want to put in. In the case of the glycans, we just define them as inert space-filling atoms that have the right covalent geometry. But one could easily extend the model if you had some feature of the glycans that you wanted to add. You could then add those features on top of the model as well. We haven't done that. We just start, again, with the simplest possible, see if there's a signature, and then try to go from there.
Thank you.
Hi. Awesome work. I know that you said that you wouldn't go over the time scale, but I was very curious. How did you reach 600 microseconds, and how did you calculate all of those?
How do we estimate the timescales?
Yeah.
Yeah, so I'm giving an entire talk about this on Wednesday that every student should attend on how to estimate timescales in simplified and coarse-grained models. So there are a lot of really ridiculous timescale estimates that are out there. Most of the papers you read, they will say something like, this 300 angstrom rearrangement takes 10 nanoseconds. And you're like, well, that can't possibly be true. And that's because people read the labels that come out of software and don't think about what they mean. So it's a great question. So what we do is we look at diffusion coefficients, because these models are energetically smooth. So things will diffuse faster than they should, right? That's just a feature of the model that we're not concerned about. We're trying to look at the overall characteristics of the energetics and how that dictates dynamics. So with the idea that the diffusion coefficient is what's wrong, like necessarily wrong, we then we compare the diffusion coefficients in our models to diffusion coefficients from experiments or from explicit solvent simulations, because they have the same length units, and then that gives you what the effective time unit is. So when I was saying 600 microseconds, it was after that conversion, which I think it's like one reduced time unit's like 100 picoseconds to a nanosecond for many systems, but that's how we do it.
Thank you.
More questions? Oh, it seems that's all then.
Okay, thank you.
Well, so let's have a applause for Paul. And then tomorrow is going to be our last day with that change that we already explained.